Hello, everyone, and welcome today to today's live broadcast and panel discussion on complementary versus companion diagnostics. We have panelists today, Martin Shaw, who's the Business Development Manager at Aerosol, and Professor Thomas Webster, the Department Chair of Chemical Engineering at Northeastern University. I'm Dr. Annette Gilchrist from Midwestern University, and I'll be your moderator for today's event. We're delighted to bring you this educational web seminar being presented by LabRoots, the scientific social networking website. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the panel discussion. To do so, you're going to click on the Ask a Question box that's located on the far left of your screen, and you're going to type your question into the drop-down box that appears. We'll answer as many questions as we have time for at the end of the presentation. If you have any trouble seeing or hearing the presentation, click on the Help Desk button that's located in the promotional board at the bottom center of your screen. Alternatively, you can just ask a question in the Ask a Question box and let someone know that you're having a problem. Any questions that we don't have time for today will be followed up with via email. Our discussion is educational and thus offers continuing education credits. You can click on the Continuing Education Credit tab that's located in the top right corner of the presentation window, and you're going to follow the process to obtain your CE credits. For now, though, please join me in welcoming our panelists. Perhaps you would each like to introduce yourself and tell us what your interest is in diagnostics, complementary versus companion. Dr. Webster, maybe you would start.
Sure, and thank you for this invitation and opportunity to share my thoughts. So I'm currently, as was mentioned, the Department Chair of Chemical Engineering here at Northeastern University in Boston. I have spent maybe about 20 years looking at nanotechnology and how nanoparticles and other nanotechnology approaches can be used to both diagnose and treat different diseases. And one of those, of course, has been cancer. So I'm very interested in this discussion of companion versus complementary diagnostic tools, since I do see a very interesting future in the diagnostics area for different cancers, as well as infection and other diseases.
Mr. Shaw.
Okay, my name is Martin Shaw. I'm Business Development Manager of Aerosol B. We develop novel biomarkers for cancer studies. I've been involved in the development of biomarkers for about 40 years, right from the very early days of immunoassay. And I followed along. So I've got a lot of experience in this, mostly in what you would call the field of complementary biomarkers rather than companion biomarkers. But both have got their place, and it's an exciting time to be in the laboratory development field. Thank you for inviting me to take part.
Well, thank you both. So I'm actually going to start with you, Mr. Shaw, and ask you perhaps if you would mind defining complementary diagnostics, how they differ from companion diagnostics, for those of us who don't know.
I think there's a difference in registration. In a companion diagnostic is registered together with the drug for which it's appropriate, and it's linked rather closely to a particular drug. It is generally expected to be compulsory to assay a companion diagnostic before you use the equivalent drug, although this is not always done. Complementary biomarkers are tests that are considered to be useful in the choice of drug, but not essential for its choice. I think that complementary diagnostics is a bit of a loose term, because a way taking a patient's temperature is a complementary diagnostic. So it's just about every lab test. But it's become to be applied more to the more recent proteomics, genomics derived tests rather than traditional laboratory chemistry. So I think that's basically the difference between them.
Great, thank you. So I think that that's important, right? The difference between them, the regulations that I'm sure are required for one versus the other, that's going to ultimately, I think, open the field up for the complementary diagnostics, right? Isn't the idea that they'll really be able to have more of them because there's less requirements that underlie their use and when they can be used and when they can't be used?
Yeah. I think it's you'll find them. I think that one of the things about complementary diagnostics is that they're likely to come from unexpected places, and that you might find that a laboratory test which was not designed to study a certain disease is found to have a value there. Complementary diagnostics have been around for many, many years, like cytochrome P450s. They've been around for don't know how many years, and they really are the original complementary diagnostics, but they're not considered so in the sort of field that we're talking about now. Things like testing for warfarin use, you really should do a complementary diagnostic, which is the P450 status, but it's not done. And probably today, if warfarin was launched, P450 would be a companion diagnostic.
Right. Right, certainly. Thank you so much. So, Dr. Webster, do you think you could provide some more examples of complementary diagnostic versus companion diagnostics?
Sure, yeah, and I definitely appreciate the thought process that perhaps we have more complementary diagnostic tools than maybe what we're acknowledging. So I think a great example of companion diagnostics is really the HER2 breast cancer diagnostic tool that I believe was, approved in the late 1990s. And there, of course, the idea is very traditional, right? You identify a marker on a cancer cell that might be exclusive to a subpopulation of patients. And in this case, it was the human epidermal growth factor receptor 2 that was exclusive to a subpopulation of breast cancer patients. And based on a positive diagnostic test, you then have approval for a therapeutic agent that's specific to that molecular indicator. So I think that's how a lot of us think about diseases and diagnostic tools. You test for a specific receptor. If it's there, then a drug follows for approval of use. Where sort of the controversy, I suppose, comes into play is comparing that with these complementary diagnostic tools where you don't necessarily need to do a complementary diagnostic tool in order to prescribe a drug for a disease. So it's recommended, just like the term says, it's complementary. So it's recommended, but it's not required. And I think and another example of a complementary diagnostic tool that a lot of people discuss is really PD-L1, right, which is the programmed death ligand 1, which is used for different cancers, from melanoma to small lung, non-small lung carcinomas, where, again, you could have a diagnostic tool or not and end up prescribing a certain drug in the complementary case.
So I actually talked about those drugs this morning, those PD-L1 monoclonal antibodies. I think another great example of the companion diagnostics really has to be the ALK-EML fusion. Again, for non-small cell lung cancer, where only a small percentage of patients actually have that mutation, and the drug itself, which is targeted against ALK, is certainly not going to be useful unless the patients actually have that mutation. So that makes right sense, right? If you have a known genetic difference where the drug is going to be useful or not useful, in the case of the BRAF, right, inhibitors, right, you really have to have the mutation or the patients actually don't do as well, that it makes perfect sense then that those would have a companion diagnostic that is required. But I do think that we have a lot of information to be gained by companion diagnostics. It makes perfect sense, right, where more information is useful in trying to decide between perhaps which therapy to use.
Absolutely. In fact, a great topic to think about when comparing companion to complementary is I think there are many people who believe that going down the road of companion diagnostics really gets you to more personalized medicine. And this is a great thing to think about is, does it really get you to more personalized medicine or not? But the idea among many in the field is, in order to make headway in cancer and some of these other diseases, you really have to personalize the diagnostic tool as well as the treatment to those specific mutations that you're talking about. So if you can personalize medicine, then I think there's a hope that it would be more effective. So by linking a diagnostic tool with a treatment, a therapeutic treatment, you can make an argument, I think, that it is more personalized than not requiring the diagnostic tool to be used, as in the case of a complementary treatment.
Yeah, I would say the other problem is once they become a companion diagnostic and they're required, right, you run the risk of, for example, insurance companies not covering if you don't have, for example, the mutation. But they know with like the estrogen receptors that sometimes patients will respond who have estrogen receptor negative cancers to, in fact, estrogen inhibitors. And so, you run that risk, right, that you make it required, but it might still be a useful drug for that patient.
And I see in the UK is demanding that some drugs are only given if there's a positive companion stroke complementary test because of the enormous costs of some of these modern immunotherapies.
Right. So we're going to come back to you, Mr. Shaw. Do you think that the distinction then between the complementary diagnostic and the companion diagnostic is important? Do you think that it's good that we have both types?
I don't think it's important very much. I think sometimes it's a commercial decision. If you are developing this new super expensive immunotherapy, you've got a companion diagnostic which you also control, then you have a commercial advantage. Whereas if you're just taking, we'll run a serum creatinine because we want to see the kidney function, there's really not much more money in it for that. So I think there is a difference, but I suppose there's no reason why a pharmaceutical company should not develop a complementary diagnostic. Particularly, there's a lot of crossover between the ownership of, like, I'm not promoting or criticizing, for example, Abbott and Roche have both got huge pharmaceutical and huge diagnostics divisions, so they're in a good position to do this. But I think the thing is that complementary diagnostics are easier to launch, and I think something that's, as a lab person, I'm a bit dubious about is there seems to be very little qualification of the laboratory performance of these complementary and companion tests. If you've got an immunological drug and you're doing an immunoassay for it, you can get apparently identical results when you run control samples, but then when you get out into the clinic, you find there are some people who react very differently. This is true about every immunoassay. So I would wonder sometimes, why don't the pharmaceutical companies say you can only use antibody A, B, C or so on? I don't think they're allowed to, because it would be restricted trade. But if you're looking at a biological, that could be important. And when you get a Me Too biosimilar and you're still using the same PD-1 antibody screening, is it the same? Having done many immunoassays, I know that this is not always the same. It might be only 1 in 100, or it might be 20 in 100, but you use anybody's developed an immunoassay has seen this.
Yeah, so I think these are great points, right? And you could really drive yourself perhaps a little crazy when you start thinking from a company viewpoint. And we've started a couple of companies in this area, so I've been in these conversations of which one is more economically beneficial for you, complementary versus companion. And certainly the points that were just raised really highlight that controversy, right? If it's linked to if your diagnostic tool is linked to your therapeutic, well, that's great commercially, right? May not be the best for healthcare, but it's certainly great commercially. But similarly, on the other hand, for complementary, if you can have, if you have a therapy that doesn't rely on your diagnostic kit, then how commercial successful can that be? So I think there's really a very interesting push and pull from the economics. I think one area, though, that we all should be conscious of is the field of theranostics, right? In the field of, bless you, in the field of theranostics, really it's one material, and many times a nanoparticle, that's combining the diagnostic with therapy. So in the field of theranostics, the two are linked, and you have the same material that's really doing both. So if that field continues and people continue to show that you can functionalize a nanoparticle with a specific ligand for, a cancer cell receptor, and then that material treats that cancer cell, kills the cancer cell, are these tools mute? In the future, what is the role really for a separate diagnostic tool? And that's, something very interesting to think about.
Yeah, I think that's a great point, and it's not even one that we have to discuss in the panel, but I think it makes absolute sense to talk about, right? Because they're completely driving some of the new technology with these theranostics, right? I mean, there's quite an interest where you can diagnose and treat at the same time.
And it's not just, the individual nanoparticle that people are looking at, but there is a growing implantable sensor community that is, again, at the same time you diagnose, you have a therapy. There's a lot of this, of course, in type 2 diabetes, implantable sensor activity. But it really, does highlight if these distinctions going to be important in the future between companion and complementary.
Yep. My apologies. I have no idea what's in the air in Chicago, but something is clearly here. So, do you think, Dr. Webster, that you could follow up with that and maybe talk about the difference between complementary and companion diagnostics and how that correlates with actually some of the FDA requirements for what is required versus a recommended test?
Yeah.
Is it a one-on-one in terms of how those work?
Right, and this is actually very interesting, and this is excellent that there's the FDA versus what's going on in Europe in terms of these diagnostic tools, right? So, as we discussed in the companion diagnostic tool, that is approved by the FDA, and then the follow-up therapy, of course, is approved by the FDA as well. So in Europe, it's different, right? As I understand it, in Europe, the diagnostic tool that's used in companion therapies or any therapies are just CE marked, right? So they're deemed safe to use, but they don't carry an FDA approval stamp as what occurs in the U.S. And again, as with everything, there's sort of pros and cons to that. It definitely, I think, makes less of a link between that tool and the diagnostic tool and the eventual therapy. In the FDA system, they are linked. And you have a company that come forward and obviously want to develop a diagnostic tool and a therapy to be FDA approved. But if the diagnostic tool is CE marked, is that linked as much to the therapy is an important consideration.
Well, at the moment it isn't, but there's a discussion paper that's been sent out by the European medical authorities, which will come closer to the American standard. It won't be exactly the same, but it's very similar that way. So they're going to be, currently they're registered as low-risk products, just like a thyroxine assay or something like that. But there will be the registration requirements will be changed, so they'll become a high-risk diagnostic, such as like doing an HIV test. So that will come pretty closer to the American system. Not the same, I can't remember what the exact wording is, but it's not the same wording, but it's very similar in application.
So along those same lines, Mr. Shaw, do you think that the type of test and whether it's actually standardized should make a difference between whether something is complementary, companion? Are they typically antibody assays? Are they typically IHC assays? Does that make a difference?
Well, I think companion assays are really all coming out to be genetic or antibody based, because you're trying to get so specific, and it's a lot to do with personalized medicine. But there's maybe losing the point, but there's no reason why they should be. It could be an enzyme activity assay or something like that. I wonder if I lost your question there a bit. I'm sorry if I may have done that.
That's okay. Do you have anything to add on that?
No.
Professor Webster?
Just yes, I would just add, and I'm sure Martin would agree with me, that I think standardization is pretty important in all of this. So I think standardization for any of these assays, these diagnostic tools that can then link to a therapy is pretty important. It's pretty important to compare apples and apples for the research field. It's pretty important to be treating patients with a high degree of standardization.
Yeah, this is there's two ways to go. The more standard it is, the fewer patients you have.
That's true.
The other thing is, the more standard it is, the more control the person who has developed the companion pair of drug and test has. And it could even get to the state of being a monopoly situation, a restraint of trade. If you said, as I think it's a great idea, it's got to be antibody ABC with drug XYZ. Company making drug XYZ would love that. But I think maybe the Monopolies Commission would not like it.
I can certainly see your commercial bent, Martin. You're clearly coming at this from the business side versus the research side. Because I think, right, for many aspects of the standardization, it has to do with will we be reimbursed and at what level is that reimbursement, and what's the cost of the assay itself, and how does that make a difference, and can it be done by a lot of different people, or does it need to be done in a single laboratory? So.
And I think, those things are very important questions, and I think maybe we don't quite know the answers to some of those questions yet. But just to add an additional intriguing thought to this discussion is we have been talking about cancer, and certainly in the field of companion and complementary diagnostic tools, cancer has been a main driver. But if you think about what's been happening in infectious diseases commercially, we have a real issue, where many companies aren't seeing a commercial value in developing new antibiotics or new strategies to reduce antibiotic-resistant bacteria. So when you have a commercial bottom fall out of some of those companies and some of those efforts, then what's left? And of course, the reason for some of this in the infectious diseases area is because the FDA has not been approving new antibiotics because you have to show that they won't develop antibiotic resistant. But think about, the potential future of having companion or complementary diagnostic tool discussions for infectious diseases. And wouldn't it be great to have some of these diagnostic tools that could then lead to a prescribed molecule to kill a bacteria that doesn't involve an antibiotic? So just to add a different dimension, it's not always about cancer in some of these discussions. There are many diseases that are important to discuss in the linking of the diagnostic tool to the therapy.
I think in the infectious diseases, there are really complementary diagnostics like susceptibility testing, but they need to be faster and easier. And in some ways, you said, get into these new antibiotics quicker. And they're going to be more expensive, so they could justify the extra cost.
So I actually think that flows nicely into the next question, which has to do with how will next generation sequencing and the decreased cost of whole genome sequencing, how is that going to play into whether you have a complementary test done? Because if you've had your whole genome sequenced, you may not need that information. for a complementary test. You may already have it sitting on a USB drive somewhere. And so the question is, do you think that that's going to matter, right? If I have my whole genome sequenced, do I need a complementary test? Maybe I already know whether my CYP3A4 is working properly or not.
Is that me or Thomas? You would like to speak?
I'm actually going to ask you that.
Okay. Well, I think the thing is, one of the points about cancers now that they're going into cheap old genome sequences, almost every even within an apparently homogeneous tumor, there is variations in genetic profile. There's also variations in gene expression depending on the blood vasculature and the oxygen tension and exposure to the drugs. So I think it's going to be that you're going to be with liquid biopsies. You're going to be screening the liquid, but the DNA in blood, and that will continuously go to new companion complementary diagnostics for a patient through the course of their disease. So it's going to. I think you're probably going to end up with a complementary stroke companion diagnostics, 1 in early breast cancer, number 2 later on, number 3, 4, 5, 6, 7 as the disease mutates and progresses and new drugs are developed. Or maybe you make a chip with all 7 on to begin with and do it is that way. But there's this enormous mutation rate of cancers is going to need to develop more and more companion diagnostics, very personalized ones.
Yeah, so I would definitely agree with that. I think genome sequencing, wouldn't that be great to have the scenario that you described where everybody's, genome has been sequenced and you can prescribe a therapeutic based on that. But, as was, I think, correctly stated, there is a dynamic part to treating cancer, right? And there are cancer stem cell research that's going on, right? There's drug chemotherapeutic resistant cancer cells that develop based upon your prescribed treatment. All of these things are very dynamic. So I think it's a one solution, fits all is certainly not going to happen. But even after having genome sequencing, I think you still have to have variability. You still have to understand that dynamic process to have a really efficient treatment strategy.
So I think in essence, you just answered one of the other questions, which had to do about the genetic heterogeneity of tumors, because that's been a clear issue that I don't know that we really appreciated 10 years ago, right? But as we've sequenced more of the cells, when you can sequence individual cells, you recognize that there are differences genetically from one cancer cell to the next cancer cell that's sitting right next to it, right? And so some of the cells may express some genes and some may not, right? And so how you treat that cancer, you're going for the whole population that you're treating, not just one specific cell. Yeah, so I think that's, so it's an excellent point.
And we need, of course, a lot more research on the heterogeneity of tumors. What is the communication between these heterogeneous cells? I mean, there's so many issues that are involved in that. And that's why really, although you have success stories of using one single drug on a tumor, how do you use one drug that can fight the heterogeneity of a tumor? It is a critical question. And that's where we really could start thinking about adaptable materials. Again, the theranostic idea that we previously talked about, an approach that changes with the changing tumor treatment is critical, I think, in the future of some of these really hard to kill cancer areas.
And certainly the ones that have become resistant, right, because of treatment that's already been given.
Absolutely.
Yeah. So I'm actually going to end with a question that I'm not sure that you can answer, but it's one that I was curious about, and that is, how do you think that the insurance industry actually views the complementary versus the companion diagnostic in terms of their reimbursement? Do they reimburse for both? Do they recognize that both are necessary, and so they're willing to pay for those, or is it more one versus the other?
I think from Europe, it's a bit in flux, but I think the trend is very strongly towards you don't get the drug if you don't do the test. It's one thing if you're giving even like penicillin, there should be a susceptibility test. But if you're looking at an immunotherapy that can cost hundreds of thousands of dollars, that's not going to be reimbursed unless you spend on the test. And some of these tests cost $7,000 or something like that. They're quite highly priced. And I suppose the point about it is, from the pharmaceutical industry, is that it's the drug is 100,000 and the test is 7,000, and they've got to pay the lab for that, and there's losses down the line. So I think that's from their point of view. But there is going to be in Europe a strong push for the fact unless you do the test, you don't get the drug. And there's also in Europe, I know, for example, in France, when they were selling HIV testing, so HIV therapeutics, that if the drug fails, they don't pay the pharmaceutical company. And I think that's also some drugs in the UK, but I can't remember the names. So there's a mutual advantage to go into companion diagnostics. I don't know, maybe you've got more experience with the US system.
Yeah, so I can add only a little bit to that. I think certainly in the companion system, there's a history of reimbursement strategies, right? So, you have a firm diagnostic tool that's been approved by the FDA, it's standardized, and then the follow-up treatment, that is a reimbursable strategy. In the complementary case, I do know of, reimbursable, of course, strategies using complementary systems where it's really up to the clinician to make some of these decisions, and that's valued pretty highly, in a reimbursement strategy. So there are some cases, certainly the points, notwithstanding, without having a diagnostic tool, the reimbursement strategy could be a little bit more difficult. But if you rely on the expertise of the clinician to treat that particular cancer, that can be.
Yeah, I think what can be sensitive is the fact that, as you mentioned earlier on, about estrogen receptors and anti-estrogen therapy, there are a minority of people who are totally negative for a biomarker but still respond positive to the drug.
Yep.
I think it's going to be, in some ways, if the patient demands it and the doctor knows that these drugs tend to be rather not toxic compared to the traditional chemotherapy, is he going to say no, or are they going to have a fight with the insurance company about it? Because there is everybody knows false positives and false biological positives and false biological negatives that's biology.
Mm-hmm.
Absolutely. And that's what brings in your comment is very interesting, and it'll be fascinating to watch how that unfolds in Europe, that if you've developed a therapeutic agent that does not show success, is that reimbursable? That is fascinating to me. And, we'll certainly have a lot to show in the field if that bears out.
Well, I think the thing in Europe is I say I know the UK better than most places, is if it doesn't work, the drug company doesn't get paid. That's what they're trying to do to get in the price negotiations with the pharmaceutical companies. They are saying, all right, we're going to give you so many dollars, tens of thousands of dollars if it works. But if it doesn't work, you take the hit. So it's very much in the pharmaceutical companies' interest to okay, there has to be some failures. Otherwise, you're missing people. But they want to keep a balance economically between treating patients who don't respond and getting all the patients who do respond. So that means there will be patients who are treated unnecessarily because their picture is such that the doctor thinks it's worth a try.
Yeah, it's a fascinating idea, and has huge ramifications for these drug companies, right? And how do you develop strategies to bring a drug to market? I mean, there's a lot of really fascinating things to keep an eye on if that,
Well, this is something that's been discussed in the industry. The fact is that you do your pricing strategy based on the fact that a certain percentage of your patients will respond. And you price the drug accordingly so that, like in the UK, they have a quality of life years, and I think it's something like £60,000. That's about $80,000 a year. I may be wrong there. Don't quote me. So they may put the price at a certain level, that including the ones who do not respond. And I'm not saying the drug doesn't work. This is biology. The the quality of life year on average for all the responders and non-responders works out at $80,000. But if you can make it worth $90,000, then you'll get a positive from the authorities. It works out, adding all the pluses and minuses, it's $70,000. Sorry, if it costs, I got it exactly the wrong round. If it only costs $70,000 for the quality of life year, you're great. And if it costs $90,000, they may ask you to trim your prices.
Yeah, I think that's a fascinating idea, right? When I think of like the ipilimumab for CTLA-4 that's given for melanoma, but only 20% of the patients actually respond to it. But those that do, right, they have a much better survival. And so you know what happens for the 80% that don't, right? I mean, that's just amazing to me, that idea that they wouldn't get reimbursed. The pharma companies wouldn't. I can only imagine how much more expensive that those drugs would become. So
we'll keep a close eye on that, right?
Yeah.
Well, watch Europe, watch the NIC. There's on their website and this sort of thing about the discussions they have, and it tends to get into the popular press as well.
I'm fascinated. So I certainly want to thank both of our panelists for their participation today. I'd also like to thank LabRoots for making today's educational discussion possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through the end of May. You'll receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's event. And that's all for now. We thank you for joining us, and we hope to see you again soon. Goodbye.
Thank you.
Goodbye.
Thank you.
Thank you.